Good evening to our principal, Dr. Anita Kanwar, our speaker, Dr. Prajakta, and all our participants. Microbiology Department of VES College is happy to host our third webinar of Inspire Me series. The last two webinars has been very enlightening, and we hope that it must have given some food for thought. These webinars are conducted with an objective of connecting with the scientists who are pursuing active research so that the participants receive the latest knowledge in those fields. We believe that today's education is tomorrow's future. I now give the floor to our Vice Principal and Head of Department, Shantanu Ma'am, to deliver the welcome address.
Thank you, Ketki. Honorable Principal of VS College of Arts, Science and Commerce, Dr. Anita Kanwar. Invited speaker of the webinar today, Dr. Prajakta Oak. Faculty members of microbiology from our college and colleges across Mumbai. All our esteemed alumni of microbiology department and dear student participants from far and wide. Good evening and a very warm welcome to each one of you. A special welcome to our speaker for today, Dr. Prajakta Oak, who has joined us from North Munich, Germany. Prajakta is also a very prominent alumnus from our department, just as was Dr. Subhojit Sen, who presented a highly enriching session last week on epigenetics. Prajakta, I'm sure you will be joining in our happiness to know that all your teachers who have taught you along with me have joined us for this session today. On behalf of the department and our entire college, I express my heartfelt gratitude and extend our greetings to all of them. Our honorable former principal, Dr. Jayashree Fadnis ma'am, and all our honorable former HODs, right from Dr. Maitri Paul, Dr. Himavadarajan, and Dr. Nagesh Malik, all of whom have been the pillars of our department. We are extremely grateful to them for being with us today. I'm sure all of them would fondly remember teaching Prajakta during her graduation and post-graduation days. We remember again with pride the fact that Prajakta had brought home the unique distinction of being the second ranker at the University of Mumbai final examinations at the BSc level, and adding to this glory and of the college when she bagged the top position at the University of Mumbai at the MSc examination too. We are really proud of you, Prajakta. I'm very sure all her teachers, including me, become nostalgic thinking about her very active student days where she would be full of enjoyment, discipline, dedication, and it was little wonder that she was also awarded the coveted best student of the college during her graduation days. Her drive for excellence was revealed to me when she, on her own initiative, had applied and was selected for a 3-month training at the prestigious NCCS Institute at Pune during her college days. The Microbiology Department of VES College of Arts, Science and Commerce takes pride in organizing the third webinar today in the Inspire Me webinar series. I am very glad to have the wholehearted cooperation of all our young staff members who, along with me, are being challenged continuously to gear up so as to keep the teaching learning process on and continue this conduct of webinars on topics relevant to microbiology and the current scenario. Both the initial webinars in this series on arboviruses by Dr. Finney Varghese and on epigenetics by a prominent alumnus, Dr. Subhojit Sen, were very well received and appreciated with over 500 participants on each occasion. The positives for us, for all of us, dear participants. I sincerely hope that all of you would benefit from today's learning exercise. And I'm sure this webinar would also be a most enjoyable one, knowing Prajakta's teaching and confident presentation skills. The microbiology department at VES College, over a period of more than 4 decades, has seen a vertical growth, offering courses right from BSc to MSc by papers, MSc by research. And since the year 2015, our department has got recognition from the University of Mumbai to offer PhD course in microbiology. Currently, we have 7 PhD students who are continuing their research under Dr. Nagesh Malik, our former HOD till last month, who I'm sure will enhance the research visibility of our department. I must agree that with the strong support received from our VES management, especially our trustee in charge, Advocate Dr. Sri Lakshman Kanal, and the vibrant leadership provided by our principal, Dr. Anita Kanwar, and of course, the zeal of all my colleagues are definitely helping us to gain confidence in conducting these webinars. We are slowly getting back to the new mission begin again that has been adopted for the present. We have to start adopting new ways and strategies, not just in the field of education, dear participants, but in every sphere of life. I would like to quote Madame Curie, who said, and I quote, "Nothing in life is to be feared. It's a time to understand more so that we may fear less. Remember to keep your spirits high and remain mentally and physically fit. Be tough, stay safe, stay healthy, stay blessed. Thank you to one and all.
Thank you, Shantani ma'am, for those encouraging words. May I now call upon our respected principal, Dr. Anita Kanwar ma'am, to address our audience and give them brief insights about our institution.
Good afternoon, everyone, and thank you, Shantini Madam. Our esteemed guest and speaker for this webinar, Dr. Prajakta Oak, Senior Scientist and Team Leader from a pharmaceutical company in Germany. Our Vice Principal and Head of Microbiology Department, Dr. Shantini Nair, other Microbiology Department members, all invitees, and my dear students. Warm welcome to you all from Vivekanand Education Society's College of Art, Science and Commerce, Chembur, Mumbai. Since all of you are attending third webinar under Inspire Me series organized by Microbiology Department of VES College of Arts, Science and Commerce, you are already aware of our college standing that we have received A grade in our third re-accreditation cycle by NAC. We are recipient of Best College Award from University of Mumbai, and Madam has already described the activities of microbiology department of our college. Microbiology department of our college has contributed greatly to college by giving college the university top rankers. Few of our alumni members have acquired teaching positions in reputed foreign universities and center of excellence. Today's talk by our eminent alumni, Dr. Prajakta Oak, on crosstalk between coronavirus and pulmonary cells will surely provide an enriching insight for the participant in this unprecedented pandemic situation. Since she is expert on this topic and I would give her chance to speak more, and I take this opportunity to welcome Prajakta to the webinar series Inspire Me. I am sure that she is definitely going to inspire all of you. So I welcome Prajakta as well as all the other participants, our members of staff, our past HODs, our former principal, Dr. J.K. Fadnis. So welcome you all and thank you.
Hello, can you hear me?
Yeah.
Thank you, Anita ma'am, for your emboldening words and constant support. A small announcement. If anyone has questions, kindly post them in the YouTube comments section. Our moderators will pick the best questions in the end. Also, the feedback link will be displayed in the description box at the end of the session. Coming to today's webinar, it is on an interesting and a very relevant topic. Crosstalk between coronavirus and pulmonary cells, a therapeutic perspective. Pranasvedam vashe sarvam trivide yat pratishthitam matena putran rakshasva shrishcha pradnyancha videhina iti. It means everything in this three world is under the control of breath. One has to take care of one's breath just the way a mother takes care of her child. Breathing has become difficult in modern time. Air pollution, lung cancer, and airborne infections are all posing a grave threat to our lungs. For today's talk, we have an expert on lung cancer, Dr. Prajakta Oak, who is a senior scientist at Pyrus Pharmaceuticals, Germany. She will be enlightening us on various interactions between pulmonary cells, the mechanism of lung infections by viruses, and also the acute respiratory distress syndrome caused by COVID-19. I request the convener of this webinar, Suman Ganga sir, to introduce our speaker of the day.
Thank you, Ketki. Am I audible?
Yes, sir.
Okay. Well, as an alumnus and faculty member of this department, it is my privilege to introduce Dr. Prajakta O, who is a distinguished alumnus of our department. The department is proud of her and her achievements. Being a microbiologist by education and a cell or a molecular biologist by profession, Dr. Oak has more than 14 years of experience in life science. She is well versed in organization and establishment of collaborative projects, experimental designs, scientific writings, and in teaching. After her journey from VS, as said by Shantini Madam, Prajakta worked as a project assistant at NCCS Pune, where she studied cavalin 1 mutations in breast carcinomas. Prajakta pursued her research career and completed her doctoral tenure from Ludwig Maximilians University, Munich, Germany. Her doctoral work in oncology was to test novel combinatorial treatment strategies in resistant breast cancer cells. She established various cellular assays and unique carcinoma mouse models in her study. She is recipient of Scholars in Teaching Award by American Association of Research, Cancer Research in 2011. Dr. Oak further worked as a postdoctoral scientist in Helmholtz Zentrum, Munich, Germany, in respiratory biology, where her role was to identify molecular mechanisms underlying neonatal chronic lung disease. She had her own scientific projects and was a pioneer member in setting up her lab. She has mentored MDs, PhDs, technicians during her time there. She has received scholarships from American thoracic societies during this time. Dr. Oak continued her research in respiratory biology and is currently a senior scientist at Pyrus Pharmaceuticals, Freising, Germany. She is leading a team with the goal of performing relevant cell-based assays to screen therapeutic protein constructs against cancer and pulmonary diseases. She will speak more on her work and about the company in her presentation. Dr. Oak has many publications in renowned national and international journals to her credit. She has also been the Indian students representative for the Consulate of India in Munich. Apart from her research acumen, Dr. Oak is involved in public outreach events where she teaches school children and enjoys playing piano in her free time. With such an experienced research background, It is our privilege to have Dr. Prajakta Oak amongst us, wherein she will speak on the current favorite virus among the scientist community world over. Why is it important for us to understand the interaction the virus has with the different pulmonary cells? Can we stumble upon a therapeutic cure through understanding these interactions? Let us dive into this webinar and find out the crosstalk between coronavirus and pulmonary cells from a therapeutic perspective. Over to you, Dr. Oak.
Thank you. Am I audible?
Yes.
Okay, great. So first of all, good afternoon or good evening, everyone. Thank you so much, Suman, for such a nice introduction. Thanks a lot. And I would also like to extend my thanks to Honorable Principal and Vice Principal, Ma'am, HOD of Microbiology Department, VS College of Arts, Science and Commerce. Thank you so much for such a warm welcome and such an overwhelming introduction that you gave. I'm really touched. And also it's an honor to know that all my mentors from VS College are also attending this presentation and my talk. I would also like to thank all the staff of VS College Microbiology Department for putting in a tremendous hard work in connecting us alumni to all of you and conducting this Inspire Me webinar series for spreading the knowledge. And last but not least, thank you all for such an overwhelming response. So as you have heard in my introduction, I have worked in a couple of different medical fields, so right from diabetic cardiomyopathy to breast cancer to lung diseases. All these fields are medically very different to each other, but one main thing or one basic thing which is common, which is underlining all these diseases is dysfunctional cells of respective organs and an impaired crosstalk between these cells. So this is mainly giving rise to the disease. And this, of course, also plays a major role in the pandemic that is going on currently, COVID-19, caused by SARS-CoV-2 virus. And I will try my level best to explain you all what is happening during COVID-19 and why there are hospitalizations associated with this disease and why sometimes this gets so severe. So let's move on to my talk. I will just share my screen. Is my screen visible?
Yes, Prajakta, all clear.
Okay,
all
great, thanks. So welcome once again to my talk. And as all of us know, that viruses are the smallest microorganisms that are found in abundance on Earth. And as they can be transmitted easily through air, food, water, by direct or indirect contact to contaminated things, fluids, surfaces, it actually makes them a primary cause of most of the deadly diseases that are occurring worldwide now. And of course, as these viruses are easily transmitted through aerosolized droplets or aerosols or droplets, they can target our lung very easily and cause many infectious respiratory diseases. So if you check WHO listings, then you will know that 24% of deaths occur due to infectious diseases every year. And out of these deaths, almost 48% of the deaths are occurring due to lower respiratory tract infectious diseases, and most of them are caused by viral infections. The mode of transmission for these viral infections plays an important role. So it is really important to understand how these viruses are transmitted from one person to the other. So most of the times it is through droplets or aerosols. So when we are talking or we are sneezing or coughing, these droplets are produced, which mostly contains infectious viral particles. Droplets can be bigger in size, so more than 5 micrometer droplets. They normally don't travel that far through the air and settle down between 1 to 5 feet of distance. And they are remaining in air for a shorter time. And then there are small droplets known as aerosols or droplet nuclei. They can remain in air for a longer duration of time and hence can cause infection to even person who is not in the vicinity of the infected person, but sometimes far away. So as far as we know from the research that has been done, coronavirus, due to its size, is mainly transmitted through droplets. So when we are speaking, coughing, or sneezing, these droplets containing infectious virus can transmit it to the other person who is in the vicinity of these droplets. And that's why social distancing is one of the main guidelines that has been put together by many governments so that we can reduce the transmission of such viruses. Then there is direct contact transmission, wherein if you are shaking hands, for example, with an infected person who probably has used his hands while covering the mouth when he was sneezing or coughing, then these droplets are on his hands, which can be transmitted to your hands. So that is a direct contact transmission. And then there is hospital-acquired transmission as well. So in hospitals, you will find such infected people in a very close proximity. So that's why it is known that hospitals are major cause of spreading such viruses, or also in case of COVID-19. It is said that you should avoid going to hospitals unless it is really urgent and necessary. And also from indirectly from the contact to surfaces. So if for example, I'm infected and I'm coughing, then my droplets are falling on the surfaces that are near me. And these droplets can remain there for a certain amount of time. For different surfaces, it is different. And if anyone then touches such a surface and then puts his hand or that part in front of mouth or nose, then this can also be one of the transmission way of these viruses. And hence, especially for the current pandemic COVID-19, many governments have put together a set of guidelines. These guidelines are mainly pertaining to social distancing as well as maintaining good hygiene. And people around the world should take these guidelines seriously. It not only will help us for COVID-19, but these guidelines can be followed also for other seasonal flus. There are many seasonal flus going around in the world. And if we follow such guidelines, then definitely we will be able to reduce transmission of such flu or diseases, respiratory infectious diseases. So before we go into details of infectious diseases and COVID-19, it is really important to know what are different classes or types of viruses out there. And if you know the differences, you will also understand why why a type of flu is different from the other type of flu. So we start with the family known as paramyxoviruses. The most common in this family is human respiratory syncytial virus, also known as RSV. The other virus belonging to this family is human parainfluenza virus, which is also one of the common ones. These viruses are enveloped, as you can see here. They generally contain negative sense single-stranded RNA, and they cause normally lower respiratory tract infection, which lead to mortality and morbidity in children, usually below age of 6 years. And they cause common cold in adults most of the times. Their incubation period is between 2 to 8 days. And it is through G protein or HN protein in case of influenza virus that this virus attaches to the host cell or the target cell, and infectious cycle is initiated. And that's why our body is mainly making antibodies against these proteins, protein G and protein HN. So moving on to next class of virus, Orthomyxoviridae. One of the most common viruses, which is the cause of common cold that happens almost every year worldwide, is human influenza virus, and it belongs to this particular family. It is also an enveloped virus with a negative sense single-stranded RNA. The cell binding occurs usually through H protein, which is on the membrane of the envelope. This has a very short incubation period of 1 to 4 days. And that's why it is okay to not follow such guidelines in case of influenza virus, because you can come to know within 1 or 4 days of infection that you have this virus, and you can take precaution. But of course, it will be beneficial if we are following such guidelines. So this influenza virus causes both upper respiratory tract infection and lower respiratory tract infection in children and adults. Later on, we will learn a bit more about respiratory tract, what is upper respiratory tract infection and lower respiratory tract infection as well. So as of now, it is just okay to understand which sort of infection is caused by these viruses. Then moving on to Picorna virus family. One of the common viruses amongst this family or this class is human rhinovirus. Unlike the first two, this is a non-enveloped virus and has a positive sense single-stranded RNA. The cell binding is usually appearing through special cellular receptor, because this doesn't have an envelope. It doesn't have proteins like H spike protein or G spike proteins, but through its polypeptides like VP1, VP2, VP3, the process of endocytosis or engulfment by a host cell or target cell is initiated, which helps then the virus to invade the cell. Its incubation period ranges from 2 to 7 days. And as these are the proteins or polypeptides which mediate then the entry of virus into the cell, mostly antibodies are produced against these polypeptides. Then coming on to the first DNA virus in this segment of respiratory viruses, it is known as adenovirus. The family, the class is also known as Adenoviridae. It's a non-enveloped double-stranded DNA virus. The cell binding happens through special receptors on the cells called as CAR receptors, and it is mostly only causing upper respiratory tract infection in children and adults. Only below 10% cases in children are the ones having lower respiratory tract infection. It also has a huge incubation period. It can go up to 10 days, almost 10 days. Then moving on to the next class, it is Picornaviridae or Picorna viruses. And one of the viruses belonging to this class is human bacuvirus. It's not a very well studied virus, but what is known is that Picorna virus, so viruses from this particular class normally are accompanying adenoviruses during infection. And they are also causing commonly upper respiratory tract infection in adults. Now coming to the virus which has created such a havoc worldwide in current times. So it is coronavirus, or the family or class is known as Coronavirude. This is also an enveloped virus. The name corona actually comes from the word crown, because it has a crown-like structure. And as I said, it's an enveloped virus. It then attaches to the host cell or the target cell through spike protein or S protein. And it is a common cause of common cold or upper respiratory tract infection, but also 50% of the cases are there where this virus can move deeper into the lung and cause lower respiratory tract infection. It's sorry for the mistake. Its incubation period may vary from 5 days up to 14 days. So again, some viruses from this family have a huge incubation period, like SARS-CoV-2, which is now the cause of COVID-19. There are many cases where no symptoms arise up to 14 days, and that's why up to 14 days, you are infectious to other people. So most of the respiratory infectious viruses are actually zoonotic in nature. What does zoonosis or zoonotic means? So these viruses don't have human as their normal or natural host. They emerge from some other species. For example, avian influenza viruses, their natural host is birds. And normally, in their natural hosts, these viruses don't cause any symptoms. So they are there, but they don't harm their natural hosts. But when these viruses jump species, for example, in case of avian influenza, the virus jumped from birds directly to human, then these viruses are termed as zoonotic viruses, the ones who are jumping species. For example, in case of Nipah virus, also the virus originated or the natural host of the virus is bats, but then it jumped species. There was an intermediate host, and then it came to human. This holds true also for coronavirus. They are zoonotic in nature. And most of the recently emerged coronaviruses, like SARS-CoV, which was inducing then SARS disease, MERS-CoV, which was responsible for MERS disease, and SARS-CoV-2, which is responsible for COVID-19 disease, they all originated from bats. They are there in bats, and then they jump to some intermediate species, and through these species, then they came into humans. So humans not being their natural host, usually such viruses are then causing a lot of severe symptoms in these unnatural hosts. And talking about symptoms, here is the table that is compiling all the symptoms that we see due to infections caused by all types of respiratory viruses. I think only influenza is missing in this table. But as you can see that almost all the viruses are able to cause upper respiratory tract infection, which is normally common cold like running nose or choked nose. So whatever is pertaining to nose, larynx and pharynx, these are upper respiratory tract infections. Then they are also causing ear infections, which is known as otitis media. And most of them are also able to move deeper into the lungs and cause lower respiratory tract infections, especially pneumonia and airflow obstruction or wheezing. And these are the symptoms which many times lead hospitalization because there is an obstruction in breathing. And I will later on tell in my talk what causes this obstruction in breathing. But to understand what is exactly happening there, we first need to understand what is our lung, which cell types are constituting the lung, how they are communicating with each other, how they are having a crosstalk with each other to maintain healthy lung function, and then what happens when a virus is entering it. So as you can see in this figure, the upper respiratory tract constitutes nasal cavity, pharynx, which contains the nasopharynx, oropharynx, and laryngopharynx, and larynx. And whatever is below larynx, right from trachea to alveoli comes under lower respiratory tract. So it's trachea, then corona, from where the trachea is dividing into left lung and right lung. Left lung is usually has 2 lobes, and right lung usually has 3 lobes. And this trachea is then branching into bronchi. Bronchi further branches into bronchioles, and the terminal end of bronchioles usually contains the structure alveoli. So, sorry. So the part from trachea to bronchioles, or even terminal bronchioles, is called as conducting zone, because this part of lung is usually aiding in the breathing. So inflow and outflow of the air. That's why it is conducting zone. And respiratory bronchioles, which also contain alveoli or alveolar sac, comes under respiratory zone, because there the actual gas exchange happens between the blood and the lung. Now, certain cell types constitute this whole structure, which then aids in the proper functioning of the lung. And mostly it is constituted by epithelial cells. So I will go through one by one and explain what are the different cell types that constitute lung and also their function. Mostly from the part right at the beginning of nose, nasal cavity, until trachea. This is made up of goblet cells and ciliated cells. So what is goblet cells? These are the cells which, so the name goblet cells comes actually from their shape. They look like wine goblet. And these are the cells that usually secrete high molecular weight mucin or mucus that is there in our nose. And this mucus normally have glycoproteins and lysozymes into it. One of the function of this mucus is to provide moisture to the epithelium, and the other function is to trap whatever foreign unwanted particle or microorganisms that are entering our nose. And usually lysozymes that are present in mucus are then digesting such unwanted particles. And hence, these are one of the cell types which are the first line of defense for us. And their functioning is then complemented very well with ciliated cells. So the name ciliated cells, because they have cilia on top. If you can see here, cilia is a hair-like structure which is present on the cells. One cell can contain up to 200 to 300 cilia. And the main function of these cilia is movement. So when these cilia are moving, they normally then aid in movement of all the debris that are produced by goblet cells inside and towards lymphatic systems, from where these debris are then flushed out of the body. So together, goblet cells and ciliary cells form first line of defense for us. As we move further into the lung, bronchi or bronchioles, there we don't need goblet cells anymore, because mucus production is effective only in the upper respiratory tract and up to trachea. But there the goblet cells are replaced by cells known as club cells or Clara cells. So Clara cells is a formal term. Now they are more commonly known as club cells. So I have written both the terms so that you will not get confused. These are again epithelial cells. They don't secrete mucus, but they do secrete chemokines and cytokines. So these are the proteins which then are attracting immune cells or activating immune system, which can then help them to clear any unwanted particle or microorganism entering bronchi. And they are also producing lysozyme, which is then helping to digest this unwanted particle and other proteins as well. Apart from producing or secreting such proteins, they also engulf toxic gases and help in their breakdown. So these toxic gases are then not going into the lower, as we move further into the respiratory tract. Now, as you see, if we go down from trachea to respiratory bronchiole, the composition of cell is changing. In the beginning, we of course need more goblet cells for more mucus production and ciliated cells to aid the transport of debris to lymphatic system from where they get flushed out. As we move down, ciliated cells are still there because we still need the movement of cilia to clear such debris. But goblet cells are then replaced by Clara cells or club cells. And when we move further deeper into respiratory zones, we don't have ciliated cells because most of the work is done until here. But we do have club cells or Clara cells, which are then secreting proteins that are beneficial. The composition of alveolar sac is entirely different as compared to the other parts of respiratory tract. Here, actually, a major crosstalk or communication between different cell type take place, which aid then in a controlled gas exchange between lung and blood. So this is just a histologic figure so you all can understand how an alveoli looks like. So these are type 1 alveolar cells or type 1 epithelial cells. They are also known as type 1 pneumocytes. They are really thin in structure, and because they are thin, they actually help in diffusion or in minimizing the diffusion distance between alveolar space and capillary. So here is a capillary, and I will explain this in detail again later. So because they are thin, they are minimizing the distance between the gas exchange from alveolar sac to capillary and back. These are mostly structural cells, and then and they constitute 96% of the alveoli. Because they are structural cells, there is a need to have more of these cells in alveoli. But 4% of it is also constituted by alveolar epithelial type 2 cells or type 2 pneumocytes. And this you can see here. These emerging structures are type 2 pneumocytes, like here. And their major function is to produce surfactant proteins. These surfactant proteins basically form a layer on top of these cells, and it helps in maintaining the tension. And so the alveoli are having the round form and they don't collapse. If the surfactant protein is not present in an optimal amount, then normally these alveoli collapse, and then there is an imbalanced gas exchange happening. So their major function is to produce surfactants, and apart from that, they also engulf toxic gases and break them down. So these toxic gases are not able to pass this barrier. And sometimes these cells also give rise to type 1 cells, especially in case of injury, where there is death of type 1 cells happening and deficiency of type 1 cells occurring. So these cells then divide and give rise to type 1 cells, and in such a way they help in repair of injured lung. So one of the fundamental cell types in lung is also fibroblast. These are elongated cells present usually in the interstitial lining. And these are one of the cell types that are able to move throughout the lung. And this movement is very, very important for them because they help in forming structure of lung or in repair of the lung. By producing certain proteins, they produce and they excrete these proteins, and these proteins are then causing a matrix-like structure. And how do they do it? So this figure will help you to understand that. As I said, they play a major role in development of lung. So these are incomplete alveoli. So you can see here you see some gaps which needs to be completed so that alveoli is perfectly round in shape and can function in a proper way. And these are type 1 pneumocytes. These are type 2 pneumocytes. They are usually then also covered by capillaries, which has endothelial cells. Mainly endothelial cells maintain the integrity of capillaries, and they also help in exchange of gas. And you can see myofibroblasts are actually sitting on this incomplete tip. What they do by sitting here is they produce structural proteins. So there is a mesh-like matrix that is produced by these cells. So they produce the matrix here, and this mesh-like matrix then forms the basis for epithelial cells to grow. So when there is a matrix here, this epithelial cell will divide and grow, and this will help in completing the alveolar structure. This is how fibroblasts are helping in lung development. This is a picture of a normal lung, and you can see perfectly round alveoli present here. And these alveoli are then aiding in gas exchange. What happens during injury is that some of these cells die, and mainly they are type 1 pneumocytes because they constitute 96% of the lung. And when they die, you have these sort of incomplete alveoli. And then fibroblasts, which are then sitting in these interstitial lining places, they migrate towards the injured area. They again secrete their structural protein scaffold, this matrix, which helps the survived cells to grow and to form a complete alveoli. So they have a major role to play in lung development. They have a major role to play in lung repair. And also one of the scaffold or structural protein is elastin, which then is giving the elasticity to the lung, because when we are breathing, our lung is expanding and contracting. And while doing so, it can happen that all these cell types in the lungs are damaged. And to reduce this damage, we need elasticity to the lung. And elastin protein produced by fibroblast is then giving this elasticity for normal breathing process. So, as you must have known by now that the crosstalk between all these different cell types, or the communication between all these different cell types, is really important to maintain a proper structure of the lung as well as proper functioning of the lung. What happens when there is an infection or when a virus enters in the lung? So in normal cases at early stages, certain immune cells like macrophages, they come into the lung. Some of them are already present in the lung. These are known as alveolar macrophages, but other macrophages then come through this capillaries into the lung. Also, other cell types from immune system like T cell, then they come into the lung, and together they are then aiding the viral clearance from this lung, and then we recover. So in normal cases, if the immune system is regulated in a proper way, we don't get really severe symptoms, but we do get some symptoms like common cold, and then these symptoms are recovered because immune system is functioning properly. But in some cases, when there is a dysregulation of communication between these immune cells, what happens is that the immune response is hypersensitized. So there are a lot of cells coming into the lung and a lot of activity happening with respect to immunity, and this activity may cause then damage to our own lung instead of causing damage to the foreign particle that has entered. So one of the crosstalks or the communications we saw was between different cell types, which is important for proper functioning of the lung. The other communication or crosstalk that plays a major role, mainly during these respiratory infectious diseases, is the crosstalk within or the communication within the immune cells, and also between immune cells and cells from the lung. And it is also important to understand this crosstalk before we go into details of COVID-19 disease. So what happens, as I said, when any foreign particle or virus is entering our respiratory system? There are certain cell types like macrophages, which are already present in alveoli or alveolar region of our lung. Then there are other cell types like natural killer cells or dendritic cells that pass through the capillary and come inside the lung. And why this influx is happening? Because when the normal cells of the lung get infected, they give signals to immune system so that immune system can get activated. And these signals are then activating these cell types. And these cell types then recognize either the infected cell, or they recognize the virus particle itself or the foreign particle itself, and then they engulf either this cell or this particle and digest it. And hence they can reduce the infection from foreign particle. And this part where the cells are activated by certain signals and then they are engulfing the infected cell or particle constitutes innate immunity. Some cell types from innate immune system like dendritic cell can cleave the foreign particle or virus that is entering and can present one of its parts or polypeptide on their membrane. And this cell can then communicate with a cell from adaptive immune system, mainly T cell, via this small part of the foreign particle, which then it presents to the adaptive immune system, and hence these cell types are called as antigen presenting cells. So this part that it extracts from the foreign particle or from virus is called as antigen. So when this antigen is presented to T cells, they get activated, and either they can form or they can differentiate into helper T cells. These are normally called as CD4 plus cells because they have a protein CD4 in their membrane present. And these helper T cells can then produce different proteins known as cytokines or chemokines. This act as signal proteins to activate other cell types of immune system. So they can either activate pro-inflammatory cytokines, which then signal back to macrophages, natural killer cells, and all these cell types. And then these cells can, again, cause phagocytosis and clearance of virus through this particular pathway. Or these cells can also communicate with B cells from adaptive immune system. And then these cells are also presenting this particular antigen to B cells. And then B cells in turn differentiate into plasma cells, which are then producing antibodies against this particular antigen. And these antibodies also can help in viral clearance from the lung. And on the other hand, these B cells can also be differentiated into memory B cells. And memory B cells can stay in our body for a longer time. And they are then producing antibodies whenever this virus is again infecting us. And that's how we become immune to a certain virus after respiratory infection, after we once have this respiratory infection. So there is always a balance that needs to be maintained, because if this whole immune system becomes haywire or it starts becoming hypersensitive, then it can also cause damage to our own lung. And this balance is maintained by pro-inflammatory cytokines and anti-inflammatory cytokines. So these anti-inflammatory cytokines from certain T cells, they give signals to different immune cells to stop their activity after the body is recovered significantly. So this is one of the communication cascade or the crosstalk, which is also important, not just in respiratory diseases, but also in other diseases. This plays a major role. So understanding our immune system really holds the basis for many different diseases. And now coming to the third communication type, that is communication between lung cells or epithelial cells and immune cells. So once a virus is infecting our respiratory tract, there are different phases that occur during this respiratory diseases. First phase is the early illness, where virus is attacking epithelial cells from the respiratory tract. These epithelial cells are then giving signals to nearby alveolar macrophages, which are already present in the lungs, and they get activated, and then they are engulfing either the infected cells or the virus particle itself. This is the early illness phase. Many times it happens that we recover at this stage itself. It really depends on the number of virus particles that are infecting you and how effective is the innate immunity of your body. So in the late phases, if the virus still persists and these signals are becoming stronger and stronger, they also lead to communication between antigen presenting cells and adaptive immune cells like T cells and B cells. And this adaptive immunity then leads to generation of antibodies against this particular virus. So these antibodies then go and bind to the virus, neutralize the virus, and also together with the innate immunity can cause clearance of the virus. And many times we can also recover from late illness phases if this whole cascade is happening in a balanced way. But there can be some cases where this crosstalk or the communication between all these cell types is dysfunctional or dysregulated, and then it can cause acute and chronic diseases or chronic phases of the disease. So what happens here is antigen presenting cell is then producing a lot of cytokines to communicate with T cells, and these T cells produce a lot of pro-inflammatory cytokines as shown here, or pro-inflammatory proteins, which then keeps on the innate and the adaptive immune system running on its toes all the time. And this can lead to lung damage. And in chronic phase as well, these cell types are communicating with each other a lot and producing a lot of pro-inflammatory proteins as compared to anti-inflammatory proteins, and this whole dysregulation of the crosstalk then leads to damage in the lung. So after telling you about all these crosstalks that are happening within the lung, I think it is time now to move to our main topic, like how SARS-CoV-2 virus is causing infection in our lung and what causes hospitalization amongst these patients. So as I described earlier as well, this virus has S protein or spike protein on its membrane. It has two variants, spike protein S1 and S2. S1 is the protein that is then attaching to ACE2 or the enzyme ACE2, which is present on epithelial cell membrane. And by attaching here, it is then initiating the influx of the virus or the endocytosis of the virus. The spike 2 protein also helps in endocytosis of the virus, but its major role is to fuse with this endocytic membrane within the cell and to release the viral RNA into the cell. Once the viral RNA is inside the cell, it starts replicating. And during this whole process, there are many different signaling molecules that are produced by these diseased cells. And some of these signaling molecules or cytokines can cause pyroptosis. So pyroptosis is something similar to apoptosis, where the communication between immune cells and epithelial lung cell is causing a programmed death of infected cells. But pyroptosis also has an elevation in certain proteins, which then gives rise to fever. And that's why we are getting many times fever during such respiratory infectious diseases. So so viral DNA replication is happening. It is then leading to this cytokine signaling, and this cytokine signaling or protein signaling is then helping communicate this disease cell type to the immune cell types, and then viral clearance can occur. So there can be two things that are happening. One thing is that this response is controlled. Usually that happens in a healthy individual. There is a healthy immune response. So macrophages come into picture, they engulf infected cells, or then also there are antigen presenting cells which are presenting the antigens to T cells and then eventually to B cells, and then antibodies are produced. And these antibodies then go and bind to S1 protein or spike 1 protein of this virus. And this here, the tail of the antibody is called as FC part of antibody. And this FC part is usually recognized by macrophages. And then macrophages attach to the antibody, which is in turn attached to virus. And this helps then the engulfment of virus by macrophages. And this is how they phagocytose the virus particle and help in their clearance. On the other hand, CD8 cells, or okay, I think I forgot to mention about them when I was talking about immunity. So there are two types of cells. One are CD4 cells or helper T cells, and the other are CD8 cells or cytotoxic T cells. Because these cell types have CD8 protein on their membrane instead of CD4 protein, they are also called as CD8 plus T cells. And these cells normally directly communicate with the diseased cells, and then via this communication, they activate a programmed death of these disease cells. And that's how they help in clearance of virus. So on one hand, we have cytotoxic T cells, which are then helping to clear the virus. On the other hand, we have helper T cells, which are communicating with B cells, producing antibodies to clear the virus. So this is all is happening in a healthy individual with a healthy immune response, which then helps in recovery of the patient from COVID-19. But what is happening in severe cases or in people who have dysfunctional immune response or immune system, that this immune response is escalated. So there is no control between the communication, and there is a production of a lot of pro-inflammatory cytokines or pro-inflammatory proteins that are then attracting more and more immune cells in there. And sometimes it can happen that this response is also causing death of healthy epithelial cells, which are not infected by the virus. And in doing so, they are also then attacking our own lung and causing damage to our own lung. And this then causes the severe symptoms that we are seeing in COVID-19 patients. So this figure will help you to understand what is happening in the severe cases, which is normally pneumonia or acute respiratory distress syndrome. So on the left-hand side, this is how a normal function works. I have already talked about it. There are different cell types, also capillaries having endothelial cells. These capillaries are usually tightly regulated, and endothelial cells are maintaining their integrity, means they don't allow a lot of immune cells to pass from capillaries to air spaces. This is called as airspace. And by maintaining this balance, they are making sure that there is no unnecessary immune response happening within alveoli. But if, as I said, this immune response is going haywire, or it is really enhanced, or our immune system becomes hypersensitive, then the membrane integrity of capillaries is lost, because certain cytokines that are produced by certain cell types from immune system as well as from lung system, for example, IL-8 or IL-6, they are causing damage also to endothelial cells. And this makes way for a leakage from capillaries into the lung. So there is more and more immune activity that is happening in this area. More and more immune cells are infiltrating from nearby capillary, and this causes then filling of a certain fluid which is protein rich within this airspace. Normally there should not be any fluid present in this airspace, there should be only air present here. But because of all this cascade, there is fluid that starts entering into these air spaces. The start of this is normally associated with pneumonia. And in most of the times, this condition can be resolved by our body itself and patients can recover. But when more fluid accumulates within this airspace, and it goes to a level where not much of the air is left in the airspace, then not much of the gas exchange is happening, then it actually causes less oxygen being transported to different organs. And that is where the disease starts becoming severe. And that this is called as acute respiratory distress syndrome, which actually is underlined by defective breathing or distress in breathing, let's say it like that. And that is why patients need hospitalization, because naturally there is no gas exchange happening, so it has to be given artificially by putting them on ventilators. And in severe cases of COVID-19, where the patients reached this particular stage or severe stage where they were not able to breathe properly, they were hospitalized, they were put under ventilation, and still 70% of these cases worldwide were dying in spite of having this ventilatory support. So when I was talking to her, I came to know, and of course, these are also statistics based on worldwide hospitals. So in most of the COVID-19 cases, we see a recovery, be it a mild COVID-19 case, moderate, severe, or critical. We can see a recovery. This really, as you have seen from previous slides, a major portion of this depends on how healthy or how balanced, let's say it like that, how balanced is your immune system, how balanced is the communication between your immune system and different types of cells in the lung. It plays a major role in recovery. But it is also seen that some of the patients which had mild to moderate symptoms for COVID-19 or moderate to severe symptoms of COVID-19, they also required ICU admission at certain stage of the disease, and it was also causing death of such patients. So of course, the rate of the death is low, or the number of the cases compared to the recovery cases are low, but they are still happening, and also with the patients that had mild symptoms. And a major part is played in all this by the crosstalk that is happening within the lung. So until now, probably it is clear for all of you that in most of the respiratory infectious diseases, also for COVID-19, it is really important to have preventive or curative therapeutics, because if it goes to severe stage, then either it causes a lot of lung damage or it can even lead to death. And many different therapeutic interventions are being tested currently for COVID-19. I have listed a few, most of them, I'm guessing. So one of the The intervention is based on antibodies against spike protein, S1 protein. So there have been antibodies that are produced within labs, which then can attach to this S1 protein. And when these antibodies are attaching to this site, the virus is not able to bind to its receptor on the cell, the ACE2 receptor. And this is how then they neutralize. And as I showed earlier, this part, FC part of the antibody is then attracting or enhancing the attachment of monocytes or macrophages to viruses, which then engulf the viruses and help in their clearance. Then there are also therapeutic interventions that are being tested currently, which are blocking the proteases, which normally binds to S2 protein. And upon this binding, it's easy for virus to then, or it's aiding the virus to then release the RNA into the cytoplasm of the cells. And this receptor is mainly protease. And so if we can find an agent or an antibody which is blocking this protease, maybe this also helps in reducing the infection. Then, as I told you, CD8 cells or cytotoxic T cells are one of the cells from immune system which help or which attach with the disease cell, and then they produce signals which then provokes this disease cell to kill itself. And one of the therapies that is being tested is to increase such CD8 positive T cells by maybe transfusing blood or a part of blood from the infected and recovered person to the other infected person. It's obviously a very difficult task to do. So that's why we are not having much of a success rate over there. Then the other part is to target pro-inflammatory cytokines. So as you have seen before, these pro-inflammatory signals are one of the major players in causing severe form of disease. So why not neutralizing these protein signals? So why not producing antibodies against these protein signals? So one of the proteins that is identified is IL-6, and there are already antibodies present against this particular protein. It is tocilizumab, which is already in clinical phase trial currently. So we will come to know if blocking these signals can stop a patient to advance can stop a patient by advancing or by reducing this this particular severe signal. Also, corticosteroids have been tested, which can reduce the pro-inflammatory signals. Then there are other approaches as well. So there are approaches which pertains to repair and regeneration of injured lung. You have seen that this dysregulated immune system many times causes a degeneration of lung structure, or it causes harm or injury into the lung. And there are therapies which are being tested where people are given mesenchymal stem cells from umbilical cord blood, which is then thought to repair, which is then thought to divide and replace the damaged cells by healthy cells. So this is one of the approach. Then we can also target, for example, endothelial cells, because what is happening in ARDS is the capillaries are losing their integrity and there is more vascular permeability. And one of the factors which is causing this vascular permeability is VEGF-A. This is a protein produced by other cell types, which is then communicating with endothelial cells for certain types of functions. And there is already a drug present for cancer therapeutics, which is then reducing the amount of this particular protein. And if we can use it here, then we can reduce the vascular permeability and reduce the inflow of such immune cells within the lungs of severe severely infected patients. Also, as virus is attaching to ACE2 receptor, it is reducing the presence of ACE2 receptor on the cells, and it is also aiding in shedding of this receptor, and this receptor is involved in normal functioning of epithelial cells as well. So it has been also tested whether we can deliver ACE2 enzyme itself from outside, which can then help this epithelial cells to recover damage. So this is also one of the therapeutic that has been tested currently. And then there are drugs that are already available, like hydrochloroquine, which is in news for quite a while now. And this drug is mainly targeting the inhibition of virus-linked processes, like inhibition of replication of virus, for example. And it has also been tested, or it is being tested, if hydrochloroquine can, or such other drugs can be also important therapeutic interventions for COVID-19. But mostly all the pharmaceuticals, all the labs, or most of the labs around the world are trying to come up with therapeutics which involve antibodies for neutralization. And we have seen many reports where such antibodies are failing to do their job. They are failing to neutralize, but in fact, they kick in more viral infection, or they kick in more inflammatory response, causing lung damage. And why this is happening? So one of the mechanisms can be that the antibody which is attaching to the virus surface, spike protein, is not able to completely neutralize the virus. So this antibody still stays there. It is not recognized by macrophages and not cleared from the system. And then this antibody can attach to other cell types from respiratory system and cause internalization of such viral particles through this antibody recognition. And these internalized viral particles can then replicate, making use of host machinery, and can cause more infectious particles. This particular process is called as antibody-dependent enhancement of the virus particles, or ADE, which instead of neutralizing is causing release of more infectious virus particle. It can also happen that this FC receptor is recognized by macrophages, but instead of having controlled or balanced communication with other cell types from immune system, this macrophage is then having an enhanced communication, which causes more pro-inflammatory cytokine or chemokine release and causing more damage to the lungs. So this can be these are two hypotheses that are put forward currently for COVID-19 failures. And one of the ways that is being discussed right now of how we can reduce such mishaps with antibodies is to have another antibody produced against this FC receptor on cells, which is recognizing the FC part of antibody. So what will happen is that this FC receptor will, this block or inhibitor antibody will then block the FC receptor part from different cells, so that these antibodies which are still present in the system and are attached to the viruses will not be recognized by other cell types, and such mishaps can be reduced. This is currently ongoing. We don't know how the result of these trials will be, but let's see. So we have seen so far that there can be failures, or there are failures associated with antibodies that are majorly being produced by different labs around the world, not just for respiratory infectious diseases, but also for other diseases. Antibodies or antibody-based therapeutics play a major role. But because antibodies can cause such unwanted processes to occur, which enhances the disease rather than reducing the disease. There are other therapeutic agents as well available in the market nowadays, and these are called small molecular therapeutics. So antibodies are normally bigger in size. They are almost 150 kilodalton. But these small molecular therapeutics or small molecular substances or proteins or RNA, DNA, whatever is used in the therapy, they are almost 10 times smaller than these antibodies. And hence, they have many different advantages to be used. So I have listed here some of the small molecular therapeutics that are already available. One is gene therapy, so where you are actually injecting or delivering a specific gene, which acts as therapeutic. Then there is RNA interference therapy. So these are short interfering RNAs that are being delivered, which then go and either stops the translation of mRNA or destroys the mRNA, which is then the key player in that particular disease. Then there is aptamer-based therapy. So aptamers are really small stretches of RNA and or DNA. As I said, they are 10 times smaller than antibodies, so they have an easy access in the body. So either we can use aptamers as therapeutics for inhibition or activation of certain processes, or we can also club our therapeutic agents to aptamers for a specific delivery within the body. And then there are protein scaffolds, which are much, much smaller than antibodies, which can also act as inhibitory therapeutics or activatory therapeutics in many different diseases. And here you can see a list of different companies that are working on such alternative therapeutics, and one of them is my company, Pieris. And this brings me to the end of my talk. There are a lot of people to thank, but I would definitely like to extend my gratitude to the whole teaching and non-teaching staff of VES College, especially chemistry and microbiology department. So because of their encouragement, their appreciation, and this relationship that we are having for my entire life, I'm really touched. It helped me a lot. So I would say that VES College was my basic and first mentoring platform, and it will always be my mentoring platform. I'm really happy to be associated with such an honorable institution. And as you all heard in my introduction, I moved to National Center for Cell Science, where I was selected for vocational training. It was under Dr. Sandhya Sittaswat. Sorry, I couldn't find her photo, so that's why I just mentioned her name. She was my first mentor to introduce me to the world of cells. It was her lab where I actually got introduced to cell culture, to animal cell culture, how we can maintain it. Here I was working mainly on diabetic cardiomyopathy. Then after my master's, I, for 2 years, was also working under Dr. Kare in the area of breast cancer. And she was also one of my mentors, and I learned a lot from her. Then I moved to Munich, to Department of Pharmacy, LMU, where my PhD mentor was Dr. Andreas Reudel, and he taught me a lot about science, about projects, about how I can develop myself into a researcher. So I thank him as well. And then I moved to do my postdoc in Comprehensive Pneumology Center in Helmholtz Center in Munich. And there my supervisor, my mentor was Dr. Anna Hilgendorf, and she taught a lot about lung to me. And also we had a clinical partner, Dr. Kai Foster. He's a neonatologist, and he was the one who introduced me to neonatal care center and taught me what is happening in patients during lung diseases. So with this, we come to an end. Thank you all for such an overwhelming response once again, and I am open to take questions now.
Thank you, Prajakta ma'am, for such an enlightening talk. Your talk has given detailed information on the subject, and you have explained it in a very nice way. Many of the participants are having questions. So let's proceed to the question answer session, which will be moderated by Dr. Shantanu Nair and Dr. Donna Joseph.
Hello, Prajakta. That was a wonderful session and an excellent presentation that you have shared with all of us. You have gone into the depth of so many aspects that the questions that were coming at the beginning were being answered at the end of your talk. So we have selected some questions. We will be asking you one by one. Um, the first question that the audience want to ask you is that they have seen that corona infections appear again in recovered patients. Is this true? And if so, why doesn't the adaptive immunity of the body come into function, preventing the infection from reappearing?
That is actually a very good question. And the answer holds true not only for COVID-19, but for many different respiratory diseases, also for flu. So with viruses, they have an ability because they are integrating their genetic material into the cells. They have an ability to modify their genetic material. So it's also, it's seen that in case of normal flu virus, the change that happens in the strain that occurs every year or every season is 5%. So viruses are always adapting. And this change in their genetic material then can lead to a situation where the antibodies that are produced in our body already because of the first infection are not able to recognize that particular antigen because that antigen has been changed a little bit during this evolution of viruses. And that is why the antibodies that are produced already by first infection becomes ineffective for second infection, and a reinfection cycle kicks in. So this is also one of the major reasons why reinfection is kicking in for COVID-19 as well. And that is why there is a fear that there will be an evolved virus which might come during the next season of the flu, which is autumn now.
Hello, Prajakta.
Hi, ma'am.
Yeah, thank you for that lovely presentation. It was very informative.
Yeah, I hope it was not too much in detail.
No, not at all. I think it was a treat for everyone who was listening to it. Yeah, so Prajakta, another question that the audience has asked is, could antihistamines help in the treatment and spread of COVID-19 by remodulating the cytokines and by reducing sneezing?
Yeah, so currently we don't know if that is possible or not. But as I said, such agents like corticosteroids, even antihistamines, are being tested right now in patients. And we will see, of course, theoretically, there should be, or they should be effective to a certain extent, yes. But you never know what is happening with this particular virus and the response related to this virus. So we can only come to know after all the clinical trials and the data is out there, when the data is out there.
Yeah, hi Prajakta. It was wonderful. The presentation, the diagrams, and it was so easy to understand what happens in normal lung and what happens under stress conditions. So really, thank you so much. I know it takes a lot of efforts to make those presentations. So we move on to the one of the question which has been asked. Do immuno booster supplements really work to strengthen our immune system? If yes, how do they work? Also, does the arsenic album 30C, it's a homeopathic medicine recommended by Ayush Ministry, really work in antigen antibody mechanism as it is propagated?
So honestly, I'm not a medical doctor to answer specifically such a type of question, but I know that a lot of people around, because there have been so many messages with recommendations and so on. As you have seen in my presentation, it is really important to have a regulated immune system. Let's say it like that. So you cannot say, let's say a healthy immune system, but it's more a regulated immune system. So this regulation is... Hello?
Yeah, Prajakta, you can continue.
Okay. So yeah, so we should have a proper regulation. There should be proper checkpoints so that this immune response is being halted at a certain time whenever required. And there should also be activation of the immune system whenever required. Normally, maintaining a healthy lifestyle will have such a regulated immune system already present in your body. Immune boosters can help, but again, anything in excess is toxic. And why it is so? Because if you are taking a lot of immune boosters, you will not come to know that you have taken it in excess, but then they are boosting your immunity so much that these checkpoints or these inhibitory cross talks is getting overwritten. And so your immune system will be hyperactive, and that is also not good with respect to such respiratory infections and COVID-19, because we have seen in the presentation how this can then lead to symptoms like pneumonia and ARDS. So anything that you do should be done in an optimal way, not too much, not too less. I think the best way to boost your immunity is maintain a healthy lifestyle, eat proper food, eat cooked food, eat your food at a proper regulated timings. And I mean, we have many things in Ayurveda that, that are there that we are consuming anyway in our food that helps boosting our immunity, like garlic, ginger, turmeric, and so on. So I don't want to be the person to comment on whether you should take supplements or not. You should definitely ask your physician, because it depends on a lot of factors. It depends on your health. It depends on your history of disease. It depends on your immune system. But of course, whatever you do should be done in a moderation, not too much. Regarding arsenic album, again, I'm not sure. It's a very new concept. And I don't think we have enough data out there or enough statistical data out there to prove that this is the best way out. Of course, again, talk to your physician and see what are the recommendations. But this is a very new disease. It's not even been a year since this virus has come in contact with humans. We are not its natural hosts. So it will take time until the virus is accepting us as hosts and we are accepting the virus as well. And once that is happening, then I think we won't need to have such immunity boosters and so on. But yeah, it will take time.
Very well put in Prajakta. Very true. We need to consult our physician. That's the best thing to do.
Yeah.
Prajakta, we are moving away a little into virology. I don't know if this is your domain, but this question needs to be addressed, which says that what is causing the severity of the corona or the COVID infection? Because in many cases, the symptoms are not visualized even after 14 days. What could be the reason? What are the factors in the virus, maybe its pathogenesis, which makes it go deeper inside the lungs? Is this understood? Are there different strains of the coronavirus which cause mild or severe infections? We would appreciate your thoughts on that, though this is not virology, may not be your area. What are your thoughts on that?
Right. So as you very rightly said, ma'am, I'm not a virologist, but based on whatever knowledge I have, I will try my best to answer this question. So can we segment this question? Can you repeat it, ma'am?
Yes.
Because it was a long question. So.
I'll just repeat it for you. The question says that what are the factors in the virus which decides the severity of the infection? because
Okay.
we see that sometimes symptoms are not visualized in 14 days, some cases are asymptomatic. So are there any strains, different strains of the viruses which can, result in a mild or severe infection? So what are the factors? Primarily, what are the factors in the virus? Is it understood?
Right. So there are many different things that actually plays a role in considering the severity of the virus. Based on my presentation, I can answer it partly. So first of all, how many virus particles have you inhaled or are there in your lung plays a major role. Because to begin with, if you are in a close proximity to a person who is infected, and then you are taking in all the droplets or most of the droplets that he's producing while talking or sneezing, then it can happen that you have a lot of viral particles inside your lung to begin with, and then they are replicating and producing even more factors. And in such cases, it can happen that even if you have optimum immunity or optimum immune system, your immune system is falling short of fighting to this virus because the virus number is too much as compared to the immune cell number. So this can be one of the factors which can cause which can decide the severity. Other thing, of course, is the regulation of cells, as I said, because as you have seen in many severe cases, it's basically dysregulation of immune system that is causing those symptoms to come in. So this is another factor that is also deciding whether the disease will be severe for you or not. Thirdly, if you have any predisposing condition, so not just lung condition. I mean, for example, if you have asthma, of course, your immunity is already haywire in asthma. And that's why if you are getting on top COVID-19 infection, it will right away kick in the severe phase of the infection. But even if you have a disease which is related to some other organ in the body, such immunity-related events are already happening in your body. And so your blood is already charged up with all the immune cells. And this might be one of the reasons why people who are having already some ailments are hospitalized right after the infection. Why? Sometimes even after 14 days, we don't see the symptoms. So one possible explanation for that is that your body has an optimum immune system, and all the other factors like how many infectious particles are in there are in favor of you. And your body is able to neutralize that virus even before the symptoms kick in and you are recovering out of that virus. This is the carrier stage. But it also means, like, as I said, not all the viral particles are being engulfed by macrophages. So there can be some viral particles out there which are still present in the lung, not enough to infect your lung, but enough that they can come in the droplets and you can infect some other person who is in your vicinity with them. And that's why it is also said that carriers might be the people who are also infectious in case of COVID-19. The other possible reason why you don't see symptoms is a low infectivity rate of virus. So the number of viral particles entering your body are so less that to reach an infectious virus dose, it needs time. There can be delay in the virus recognizing its host and getting into its host, which is then further delaying the replication of viral particles, and hence it is not reaching the infectious dose. This can also be one of the possible explanations. Now coming to the question of different strains. As I said, this is very new. It has been just a few months, and we can consider this as one season of infection. I am not aware of the very latest development, but as far as I know, studies are still going on. We don't have any concrete answer on whether this is one strain or there are different strains. And of course, a lot of studies on a lot of different people from different regions need to be done to answer this question. But yes, knowing that the virus can mutate and the strain can change, people in the scientific and medical community are actually anticipating that it can happen, that in the next season, which comes towards the end of the year, we can have some different virus strain from this one.
That was a very well answered question, Prajakta. And I must add to this that people all over the world, all scientists are wondering, how is this virus, being able to work across all seasons? Okay, irrespective, we thought with the rains or with the summer, it would be wiped out. So there is a lot to know about the pathogenic mechanism of this virus.
Yeah.
Yes, Prajakta. Hello, am I audible?
Yeah.
Yes, ma'am.
Yeah. So the next question is, can coronavirus spread through feces? Now, this was an interesting question and I took the liberty to answer it. I would just like you to confirm whether it is right. What I said is that some research has found that virus particles have been shed through feces. And this is especially in people who are severe cases. But could this be a problem in a place like India where, sanitation conditions are not so good? Could it be a mode of transmission of the virus?
This is, again, a very good question. So the research that was done on SARS virus, so SARS-CoV, which is very close, it has almost, I think, 70 to 80% similarity to COVID. It shows that it was also able to be transmitted through this fecal oral route or through feces. And if someone is touching some contaminated feces, then of course, this virus was able to be transmitted like this. As you said, for SARS-CoV-2 as well, there is a lot of research going on, and there are hints that it can be transmitted through fecal oral route. It is not yet confirmed, but there are some hints. Again, this is happening in severely infected patients because severely infected patients are having a lot of infectious virus particles within their body. And maybe these virus particles are also able to infect other cell types, not just lung epithelial cells, because the major point of entry of this virus is through attaching to this enzyme, specific enzyme receptor called ACE2 receptor. And ACE2 receptor is not only present on epithelial cells, but also present on other cell types. So via this entry route, maybe it is also able to go to different parts of the body and not just is contained in the lung. We majorly see lung damage because the route of entry is through nose and through trachea, and then it is the lung is the most susceptible organ to such a virus. So yeah, quite possible, but not yet. A substantial amount of research needs to be done before we can confirm this.
Thanks, Prajakta. This could be the reason also why there are symptoms like nausea and abdominal related.
Quite possible. Yeah, even diarrhea is one of the symptoms of COVID-19, or vomiting. So it can show that virus is entering other organs as well.
Okay. Prana ma'am, shall I take the next?
Yes.
Yes. Well, I'm again back with the question, why anti-malarial drug HCQ is being used to treat a viral infection. And municipal hospitals in Mumbai are using it, even though it has been advised by WHO to stop the usage of the drug. So I don't know. I know we cannot comment on why Mumbai hospitals are still using it, but why anti-malarial drug is being used for a viral infection? That is something if you can answer on.
So malaria is also an infection caused by virus, right? And those who attended the arthropod virus seminar series, I think, got more insight into all this process.
Right.
I'm not aware of the detail of mode of action, but what I know is that drugs like hydrochloroquine are actually targeting the replication process of the virus. So they are inhibiting the replication process of the virus. And now also, if we can use such already existing drugs, because this has become a pandemic, it's a need to have some sort of curative or preventive medicine out there as fast as possible. So people are trying out all different kinds of medicines, which are either targeting the virus itself, neutralizing the virus, or targeting its replication. One of them is hydrochloroquine. So you can contain the replication, and via this mode, you can clear the virus out of your system. Or they are also targeting immune system, cells of immune system, cytokines, chemokines. So they are targeting all different ways to see.
Whatever we have in our kitty.
Yeah, exactly. And I think hydrochloroquine showed some promise in the beginning, but as it is not accepted worldwide. So for example, Europe was against using hydrochloroquine,
Right.
even if they had a lot of patients. So yeah. Okay. Maybe in past there have been cases where this drug showed an adverse effect in many different patients of malaria.
Right.
Quite possible.
Possible. Thank you. Shantri ma'am, over to you.
Hello, Prajakta. There are a lot of questions, and some of them you can mail them to you directly. But there's one good question which asks whether the damage caused to the lungs by the coronavirus, is it reversible?
Mm-hmm. That's again a very good question, actually. So it really depends on the extent of damage that is caused. So one of the damages I showed you was filling of lungs with a fluid, which is associated with pneumonia and ARDS. This damage in some cases can be reversible, because if you are fixing the leakage, then the lung is, and if your lung is healthy enough, then it is able to revive itself from this particular sort of damage. This is just one type of damage that is happening. There are many different damages that is happening due to the cytokine storm. One of them is killing or dying of normal lung cells. So most of the cells that are dying are alveolar type 1 cells, because as I said, 96% of our lung has alveolar type 1 cells. And this damage, of course, is irreversible. But if the damage is happening to a lower extent, then alveolar type 2 cells can differentiate themselves into alveolar type 1 cells and divide, and over time they can fill this gap which is formed by death of alveolar type 1 cells. The other cell types which is dying is endothelial cells, because they are the major cell types in capillaries. They are also affected by all this cytokine storm that is taking place in lung. Also, fibroblasts are cell types which are dying. So if fibroblast is dying, it is a key player in repair, as I showed you. So if fibroblast is dying, then of course it is affecting the repair process happening within the lung. And for such post-COVID damages, one of the therapeutic interventions that is being tested currently is also use of stem cells, so mesenchymal stem cells. So they are being injected directly into the lung, and people are checking whether then these stem cells are able to revive all the cells that are dead in the lung, and then can cause or reverse the damage that is happening in the lung. Yeah, so sometimes it is reversible, sometimes it is not.
Okay, thanks, Prajakta. So that was the last question. So over to you, Ketki.
Thank you, Shantani ma'am, Dona ma'am, and Suman sir for moderating and asking questions on behalf of our participants. Now I request Dr. Shweta Patil to propose the vote of appreciation.
Good evening, Prajakta. It was truly a very informative talk, and which was very much needed in this pandemic with the type of questions that we had. So on behalf of the management and VES College of Arts, Science and Commerce, I would like to thank Dr. Prajakta Oak for accepting our invitation and delivering an excellent talk on the underlying implications and therapies of respiratory diseases with relevance to COVID-19. You actually started from scratch and built so well on the cell's crosstalk in a complex organ and the changes that happen during infections. We also thank Dr. Catherine for the help rendered. Special thanks to Pyrus Pharmaceuticals. As Winston Churchill quotes, we make a living by what we get, but we make a life by what we give. It was Prajakta's wish that she should help her alma mater, and hence, amidst this pandemic, where she is actively involved in possible therapies for COVID-19, she made the time and adjusted her time frame for the benefit of the students. We thank you, Prajakta, and all the best to you. Sincere gratitude to our principal, Dr. Anita Kanwar, for constant support and for attending the sessions in spite of a busy schedule. Heartfelt thanks to Mr. Shashank Joshi for the technical support he provides continuously for organizing the webinars. I also thank the team in microbiology department at VESAC for the efforts to make all these events successful. Last but not the least, I would like to thank all the participants without whom this event would not have been possible. Special thanks to many of you who are attending all the webinars in this series. Thank you. Over to you, Kitki.
Thank you, Shweta ma'am. The feedback link is displayed, and fill the form and give your honest feedback, as it's very important for us to learn. After filling the feedback form, your e-certificate will be auto-generated. As we have reached the end of the session, I would like to refer what Rudolf Virchow said. The body is a state where every cell is a citizen. Disease is merely the conflict between the citizens of the state, which is because of some external factors. So let's reduce the conflict and increase the immunity of the body. We don't know how strong we are unless staying strong is the only option left. So let's stay safe, stay indoors, and fight this battle against coronavirus. Thank you for joining the session. We will be meeting soon in the next webinar of this series, the details of which will be announced to you in the due course. Thank you all for joining in.
Thank you, Prajakta.
Thank you, Prajakta, and bye to everybody.
Bye to everybody.
Thank you as well for inviting me. It was great.
It was great meeting you, and
Bye,
thank to all our former HODs and former principals.
Thank you.